So we are very pleased to have today with us in our Bar-Ilan University Vision Science Seminar, Dr. Michal Ramot, who is an assistant professor at the Department of Brain Sciences at the Weizmann Institute of Science. Michal did her BSc in mathematics as part of the Amirim Excellence Program at the Hebrew University. She then completed a PhD in neuroscience in the Interdisciplinary Center for Neural Computation at the Hebrew University, and she did this under the supervision of Professor Leon Duell and Professor Rafi Malach from the Weizmann Institute. After her PhD, she did a postdoc at the NIH, working with Professor Alex Martin. She publishes in top leading journals as Trends in Cognitive Sciences, PNAS, Journal of Neuroscience, eLife, and more. She was recently awarded the prestigious European Research Council ERC starting grant. She's the head of the Ramot lab that focuses on investigating networks behavior and plasticity in the human brain. Her group studies large-scale interaction that support the network-level synthesis, I'm not pronouncing this correctly, of interdependent cognitive processes such as perception, action, memory, learning, and social cognition. Her group builds and tests hypotheses about the relationship between behavior and neural network characteristics as functional connectivity and dynamics using imaging methods, electrocortical recordings from patients, objective behavioral measures, and covert neurofeedback. So without further ado, I'd like to welcome you, Michal, to our Vision Seminar to hear about your exciting research, and thank you for joining us.
Thank you, Sharon. That was a lovely introduction. Like Sharon said, I'm going to talk about characterizing the causal role of large-scale network interactions in supporting complex cognition. And okay, so when I talk about complex cognition, it's maybe a bit of a misnomer even, because there is no non-complex cognition. All cognition is very, very complex, and all behavior is incredibly complex. And I think in neuroscience throughout the ages, we've done a very good job in mapping the brain. So we know what brain regions are specialized for. We generally understand this pretty well. If you point to a brain region, I can tell you what it does. And this is really good for understanding function, like simple function. But like we said, there is no simple function. There are no simple behaviors. All behaviors require the integration of multiple different processes and multiple functions. So in light of this, I think one of the big challenges now in neuroscience is how we move from looking at brain regions to looking at networks. And one of the reasons that networks are important, or that we know the networks are important, is that we know that there are large variations between individuals in performance on pretty much any task, as well as in clinical symptoms and all this. So we have these large individual differences in behavior. And we also see that correlations in spontaneous activity between different brain regions, and also in task-based activity, but even in spontaneous activity, between these different brain regions predict individual differences in task performance and clinical symptoms. So we have a good basis to assume that these network structures are important. So this is an example of this kind of network-based activity. This is from my study I did with Alex Martin during my postdoc. And what we did here was look at face memory. Now, like I said, there are very large differences in how well people do at remembering faces. People can be very, very good face recognizers or very poor face recognizers. We have the whole gamut in between. And what we were searching for are the neural correlates of this. So what network structures explain people's ability in recognizing faces? And what this map is showing basically is regions that do that. So all these voxels, like the yellowy orange voxels, what they're showing, the value of these voxels is basically their predictive power. So these are like second order correlations. The voxels here, so for instance, if we take this here, the somatosensory is like somatosensory face area, basically. What this means is that the more strongly correlated these voxels are to the face patches, the better people are at remembering faces. It's a correlation between brain and behavior. So we take people's scores on the standard face memory test, the Cambridge face memory test, and we correlate them to the correlation between each voxel and its correlation to face patches. Okay, so what we see here in the map is how strongly predictive that correlation is between each voxel and the face patches. So the more connected the voxels are to the face patches, the better people are at remembering faces, the better they do on the Cambridge face memory test. And what we see here is that this spans a very wide network. So we have face regions over here. We also have memory areas. We have low-level vision areas, social cognition areas, of course, very broad. And when we think about the task that is face memory, then of course this makes perfect sense because we don't remember faces in a vacuum, right? We don't remember just the visual features of the face. Like when I see Sharon, then I'm not only seeing her visual features, I'm remembering her as a person or in the social context that we met, in the social context that we know each other. I might remember previous interactions we've had. I might remember the way she walks or the way she talks. So I have this whole concept of Sharon as a person. And whenever I see her face, this whole concept is instantaneously activated. So there's no way to separate just like the visual features of face from this task of face recognition. So these are correlations, resting state correlations that predict performance on the Cambridge face memory test. And you might say the Cambridge face memory test doesn't involve any of this, right? There's no social context for the faces. And yet we still see these correlations with these widespread brain areas. And I think the reason for this is that we're looking at spontaneous activity, and spontaneous activity probably reflects like the baseline of the brain, right? So this baseline is what the brain normally does. So normally you don't see isolated faces in black and white with no hair. Normally you see people in social context. So these networks are the networks that are activated by seeing a face, even when the task doesn't specifically require all these processes, still activates these widespread networks. Okay, so let's go back to the main question we're asking in neuroscience, in my opinion, in general. So when we think about what are we trying to do in neuroscience in general is really connect between brain and behavior, right? This is the correlation we want to understand. How does the brain cause behavior in a way? Like if you gave me a particular brain state, I would like to be able to tell you what behaviors arise from what the brain is doing right now, right? This is kind of the driving question behind all of neuroscience. And the way we normally study this is we manipulate behavior in some way, and then we see how that changes the brain, or we manipulate external stimuli in some way and see how that influences the brain, and then maybe in turn how that influences behavior. And we have all these correlations between brain behavior and maybe external stimuli. But really what we would want to ask isn't this bidirectional connectivity, but how does the brain cause behavior, right? This is in neuroscience is kind of the holy grail, right? How does the brain create behavior in a way, right? This is a question we're trying to answer. So to do that, what we'd really like to do is to perturb the brain in some way, change the brain and see if changing the brain changed behavior, right? That's the more causal approach than just looking at correlations between brain and behavior. Now, this is very, very difficult to do in humans because it's difficult to do non-invasive perturbations of the brain. And what I would like to suggest is that one such way of doing it is neurofeedback. So in neurofeedback, the classic view of neurofeedback is that you're translating signals from the brain to a signal that subjects can perceive, right? And I would like to suggest a different view of neurofeedback, which is that it's reinforcing target brain states that we identify in real time across large-scale networks. Okay, and what this gives us is neurofeedback not as a conscious training paradigm, but as a neural perturbation technique. It's a way of directly changing targeted brain states. So I can look at the brain in real time, identify a state that I'm interested in, and reinforce it whenever it happens spontaneously. This is very good for studying spontaneous activity because it doesn't require anything. I'm really just monitoring the spontaneous activity in the brain. And whenever something happens that I want to reinforce, then I give positive feedback. Or if it's something that I want to train away, then I can give negative feedback, right? And this is incredibly flexible because basically it means that you can feedback anything that you can record, right? So you don't need to give people strategies. You don't need to know what a particular brain state is doing. You don't need to know what would activate that brain state. You just, whenever it happens spontaneously, you give feedback. Okay, so this, again, we go back to, we want to change the brain and then see how that changes behavior. We would like to bypass these other pathways. We don't want to change behavior because that would change many things about the brain. We don't want to change external stimuli that would activate the network. We really want something that's very direct, like directly change the brain in a way that's orthogonal to the behavior that we think that the target brain state is involved in, and without activating it through external stimuli. Okay, this we call covert neurofeedback, and we'll talk about it more. But before that, I just want to introduce the general approach in the lab. We're trying to characterize behavior and individual differences, then find underlying neural networks. This is what I showed you before with the face memory paper, right? We find the networks that are correlated with the behaviors we're interested in. And then we try to perturb the networks and see if that changes behavior, okay? Okay, so before we begin this, I need to answer a few questions. Can this kind of implicit feedback learning work with real-time fMRI? This is a big question because fMRI is slow, and learning we generally think of as happening on a faster time scale. So will this work with real-time fMRI? Can we change networks in this way? And not just activations of particular regions, or like neurons in animals, they did a lot of work on activating neurons. Can we change networks? Can we change them even if they're aberrant? So even if there's something maybe developmental about the way that they were shaped, can we still change them through neurofeedback? And then will this change the networks? This is a big question. Will this change the networks cause a change to the associated behavior? So the first study I'm going to present was done actually in my postdoc with Rafi Malach before I came to NIH. And it's about unconscious learning with neurofeedback. So what we were trying to do here is to test whether people can learn without even trying to learn. So can you use this kind of covert neurofeedback in a way that's completely implicit? So what we did is we had people come into the lab and we told them that this is a study about reward. And they were going to be hearing good sounds. Can you hear? And bad sounds. Okay, and the sounds are inherently good or bad, so I didn't even have to tell you which were the good sounds or the bad sounds. You could probably figure it out. And they were also coupled with a monetary reward or punishment. So if you heard the good sound, you were winning money. If you heard the bad sound, you were losing money. So you inherently want the good sounds, right? And you want to avoid the bad sounds, but they have no idea that they're controlling the sounds in any way. And as far as they're concerned, we're just studying how their brain reacts to reward. And what's really going on, and they have to push a button, one button for the good sounds, one for the bad sounds. That's their task. That's all they do. And what's really going on is that we look at two areas in the brain, the face areas, the FFA, and the place areas, the PPA. And we divide people into two groups, the FFA good group, or the faces good group, and the places good group. So in the faces good group, people get positive feedback if activity in FFA is higher than activity in PPA. Right? And they get negative feedback if activity in PPA is higher than activity in FFA, like for a given time window. So each TR, we look at the signals and we ask, is activity in the face patches higher than in PPA? And if so, they get good feedback. If not, they get negative feedback. It also have a certain threshold. And for the places good group, it's the exact opposite, right? So they get good feedback for activity in PPA being higher than FFA and vice versa. And the question is, even though they're not trying to learn anything, they don't think they're controlling the sounds in any way, will their brains still learn to produce the activity that gives them the good sounds, that gives them the positive reinforcement? And we see that they do. So they do learn, they get better over time. But even more interesting, we see these changes in the brain. So this is what happens in the brain on the first day of training. This is showing the voxels that are significantly more correlated to the good ROI, which could be either faces or places, depending on which group they were in, than the bad ROI, right? This is counterbalanced. And there's not really very much going on. But then by the second day, and then even more so on the third day, there are now many regions in the brain that are more connected to the good ROI than to the bad ROI, right? And these regions are regions that are related to rewards, your striatum, and like all these reward processing regions, but also areas of the default mode, which are now more connected to the good region than to the bad region. And you can do the same analysis and ask, which voxels are now more connected to faces? And you don't see anything because this is counterbalanced across the participants. So it's only, you only see this change that's related to the valence of the regions and not to the regions themselves. Okay, so yes, we can do this kind of implicit feedback learning with real-time fMRI, and even when it's entirely implicit and people aren't even trying to learn anything, it still works. But can we change networks? This was still very localized, just like the particular regions. Can we change aberrant networks, and will this change the behavior? So for this is a study that I did with Alex Martin, and we took very high-functioning people with autism. And we know from a previous cohort in the lab that these regions, here in STS and somatosensory, are the most undercorrelated in ASD participants than in typically developing participants, right? And the degree of this underconnectivity is also related to symptom severity. So the more underconnected these areas are, the worse people are on the SRS, is the social responsiveness scale, right? The worse their social symptoms are. So now what we're trying to do is to get these areas to correlate more during spontaneous activity. And we have a third control region, which we chose to be uncorrelated with the two targets. So it's uncorrelated with two targets in TD participants and typically developing participants, actually a network in a way, right? So we're training people to increase connectivity between the two targets and at the same time decouple them from the control, which is more like what the typically developing connectivity looks like. Okay, so what we do is we bring people into the lab. We look at the signal from these three regions every time point. Okay, so we have three signals that we look at every time point from the two targets and from the control. And for each time point, we ask simply, what happened between this time point and the previous time point? Did the signal go up or did it go down? Okay. And we're looking for states that look like this. We want the two target signals to be moving in the same direction. So either up or down, we don't care, but in the same direction and opposite from control. Okay, so this is a proxy for connectivity. So the more they behave like this, the more correlated the two targets are and the less correlated they are to control. And when this happens, we reward through a feedback, which I'll show you in a second. We give this audiovisual feedback, which is in the form of this puzzle task. So they see this gray screen, and we tell them there's a picture hidden underneath the screen, and your job is to reveal the picture. And they say, what? We say, just try and reveal the picture. And so they're basically lying in the scanner, and they have no idea what they're supposed to be doing. But what they see is something like this. Oh, sorry. Okay, this is what they're seeing. So they see pieces of the picture come up. This is a bit sped up. It's a bit slower in the fMRI. And at the end of the run, they get their score of how many squares they managed to reveal. And they do a few of these runs. And each run, if they beat their previous high score, they get a bonus. And they get very, very engaged in this. They're really trying, but they don't know what to do. So they come up with all kinds of strategies. Like you need to look to the left of the square that's about to appear, or you need to think happy thoughts, or like all kinds of things that really have nothing to do with the correlation between STS and somatosensory, which even if I wanted to, I couldn't really tell you what they would have to do in order to get those two to correlate. So they are trying, but their strategies aren't related to the networks in any way. Okay, so will we see improvement and will we see any long-term changes? Those are the questions. So remember, this is our little network. We have STS and somatosensory here. We're trying to get them to couple more and to decouple from this control region in IPL. Sorry. And this is exactly what we see. So over 4 days of training, we see the correlation between the two targets increase. And then we have a follow-up, which is between a month to a year after the initial training. And we see that this effect lasts for pretty much almost the same, even across a month and year. And we checked dependence on time. We didn't see any dependence on time. So this seems to be a relatively fixed effect. And similarly, the target and control become more decoupled. And it reaches a floor effect, but then it stays that way and also stays that way for the follow-up. Okay, so this is looking at the ROIs themselves. But what does this look like across the entire brain? Is this something that's really specific to the areas we were training? And the answer is yes. So if we ask, we look now, we do a whole brain analysis, and we ask which regions in the brain are now more correlated to target 2 and less correlated to control. Okay, so this is target 2 right here. And we say across the entire brain, which voxels are now more correlated to target 2 at the end of training and less correlated to control. And we get exactly the voxels in target 1, as well as a few surrounding voxels. And this is a very high threshold. So this is the peak of the change. And if we relax the threshold a little bit, we see that we get the entire network. So we get all of STS, this precuneus region here. So all of this region that's connected to STS has changed as a result. So all of this is now more correlated to target 2 and less correlated to control, which is exactly what we were training. So this was very striking. And it meant that we can change these aberrant networks, but we're still not sure about the associated behavior, right? We actually measured this as well. We did the SRS before and after, but the SRS is it's a parent-filled questionnaire. It's usually asked about, you know, in the last 6 months, does your child usually blah blah blah. So it's not really meant to measure change at this kind of timescale. We did see a correlation between change in SRS and change in the behavior in the networks, but it's a little bit difficult to interpret. So what we really wanted to do is focus on this change in behavior. So for this, we went to a better, more well-defined behavior that's easier to measure and easier to measure change in behavior. And we went back to face memory. And if you remember the first slide I showed you with the face memory networks that we uncovered, so we chose 2 nodes from there, the 2 nodes that were most correlated to behavior, and the third control node, and we did the same kind of neurofeedback. And we saw the similar changes in networks, but we started this just before COVID, and then COVID. So we don't really have all the necessary controls for this, but we do have 10 participants who finished the neurofeedback. And apart from the change in networks, we also see a change in behavior. So we took people who were very bad at face memory, so at least 1.5 standard deviations below the norm. And they also have real world deficits in face memory. So they all say that they're very bad at remembering faces and that this bothers them in their real life. And what we find is that after training, so a week after training and 3 months after training, the change in behavior is more than 1 standard deviation on the test. So this is the Cambridge face memory test. We also do the Cambridge car memory test, and there we see no change in the scores. So this is a selective improvement in the face memory following the training. So this was very cool, but also, it's not enough participants, not enough controls. So now we're working on a different experiment that does something similar and asks this in a better, even better way. Okay, so now all these things I told you about, there was this paper which said basically you can't do them, right? So many of you have probably heard of this paper saying individual differences in fMRI require thousands of participants. It's very difficult to do. This paper got tons of attention. I have a lot to say about this paper, but not for now. I think what this means is we have to be very, very careful in how we ask these questions. Not that they're not possible, just that you have to be very careful. And one of the things that this led us to do is to deconstruct complex tasks, because one of my main criticisms of this paper is that the behaviors they looked at are all very complex. And if you're doing univariate analysis, you're trying to find correlation between parcels that predict fluid intelligence, and of course, you're not going to find them. You have a much better chance of finding a neural correlate of a simpler process, right? Not fluid intelligence. So how do we take complex tasks and start to deconstruct their variance into simpler processes, which we're more likely to find neural correlates for? That's one of the main directions now in the lab. And then we also have other directions that we're going in that I'm not going to discuss in this talk. Okay, but even before we begin, we have this question about brain behavior. And really, if we want to link these 2 things, we want to improve the reliability both of the measured behaviors and of the fMRI signal quality. So we've been working on both of these. I'm going to talk a little bit about the reliability of the behavior. The fMRI signal quality we've been working on a lot, especially with new different sequences and doing multi-echo sequences. If anyone is interested, then I'm happy to talk about it after, but not in this talk. Okay, so for behavior, one of the questions is that people generally assume, but is never really asked, is do participants converge to a stable mean? So do you even have a stable behavioral trait? Like, are you good at faces or not good at faces? Is this something real? Like, are you always good at faces at the same level? Or does this fluctuate? Is this maybe very noisy? Is this even something that we can measure, this like stable trait? So to do this, we administered many different tasks and many different trials of each task. This is work by my fantastic grad student, Jan Kadlec. And we asked how stable are your scores across different splits of the data, right? So we can do like a split half analysis. We take all the trials, divide them into odd even, or like randomly divide them into 2, and calculate the correlation between the trials across participants, right? So here, each dot would be 1 participant, and this is average across their one half of the trials versus average across the other half of the trials, and we can calculate the correlation between this, and this is a measure of how stable your trait is, right? How stable your behavior is. And we can do this 10,000 times, and then average and create this reliability curves. So this is like reliability is a function of number of trials. So, and the idea is that if this is a stable trait, then if we collect enough data, if we have enough trials, then as the number of trials increases, this reliability will converge to 1 eventually. But you'll be identical. If I had 5,000 trials of face memory and another 5,000 trials on you, they should be identical if that trait is stable, right? Okay, so this is a collaboration also with Professor Jesse Risserman and Catherine Walsh at UCLA. And we collected, we wanted to ask how reliable commonly used cognitive tasks are, right? So we collected data on 17 tasks from about 250 participants. They each did about 8 hours of online testing over 4 testing days. A lot of data. And we did this kind of split half analysis on all the tasks. And what we found is 2 very interesting things. So we can predict how this is going to converge. So this, we can model mathematically how reliability will change as a function of number of trials. So we can predict what the reliability will be. This is real data. This isn't predictions, but we show that we can build predictions. And all these tasks eventually do converge to 1. So it seems like they are measuring these stable traits, but there is a big difference in how reliable they are. So this is just the different measures that we're looking at. And this is the reliability of one standard administration of the task. So if you were doing the task, this is what people generally do with these tasks, right? And some of them are very reliable, some of them are very unreliable. And there's a very wide range. So it's difficult to say how reliable a task should be, but it really affects the correlation. So why do we care, right? Why do we care how reliable tasks are? Because when they're not reliable, then, and what we're trying to do is measure a relationship between brain and behavior, right? So here I'm showing a different relationship between 2 different tasks, but mathematically it's the same. So if you have 2 different measures and they're not reliable, like here the reliability of these 2, we're comparing 2 different tests. This is Cambridge car memory test and the car matching. The real correlation is very, very high. But if you take only a few trials, so now they're not reliable, then this is the observed correlations. They're going to be very low. You're not going to be able to see this correlation between the 2 tasks if the tasks are not reliable, right? Similarly, when the tasks are not very highly correlated, so this is PG&G, the parametric go-no-go, and this is a task we developed. So they're not very well correlated. But if they're unreliable, then you'll see even lower correlations, maybe even anti-correlations. You might see even correlations are a bit too high. But generally, if you see high correlations, then they're real. This is something that's, we don't get fake correlations above 0.3 or 0.4. Okay, so if you see high correlations between 2 measures, then they're very likely to be real. But if you have unreliable data, then you might not detect these correlations. Okay, so it's very important for data to be as reliable as possible. Okay, and Jan also built this nifty web app you can go to, where you put in just say you have a pilot experiment or you want to know how reliable your task is, then you put in the mean of of the performance across all the participants and the variance across participants, how many subjects you have, and it will build this curve for you, this reliability versus number of trials curve. And then you can see how reliable your data is, how reliable it will be if you add more trials. And you can play with this, and this is a very nice tool for building experiments. If you want to build a new experiment and you want to know how many trials you need for your task to be reliable, then you can use this app. Okay. Now, this was like a preamble. Now we want to get into what are we doing now, right? So I said we're very interested in behavior and building better behavioral measures and also trying to deconstruct behavior in simpler processes. And one of the things that's difficult is finding good, reliable behavioral measures. And this started when we were doing the project with autism, and we wanted a good measure of social cognition, and those are very difficult to find. They're all either subjective or questionnaire-based or interview-based, and it's very difficult to find a good objective measure of social cognition. But it's not just social cognitions, it's like many different modalities. So out of this came the motivation to try and search for better behavioral measures. We want something that's objective, right? That has sufficient variance across the population, because we want to look at individual differences. So if people are all at ceiling or people are all at floor, or there's not really any variance between people, then it'll be very difficult to tell them apart, and it would be very difficult to find correlations to the brain. We want it to be stable and reliable. And we also want it to be sensitive to change, because we want to do neurofeedback on it and then see if we want to identify a brain network that is correlated to the behavior, and then we want to train the brain network through neurofeedback and see if that changed the behavior. So the behavioral measure has to be sensitive to change. And once we have these behavioral measures, then we can find neural correlates. right? So we started looking at eye movement typicality. So eye movements are a very, very rich source of data, but they're very difficult to analyze, because it's very difficult to know what should be, right? So for still images, it's a bit simpler, but for movies, how do you know how do you score eye movements on movies? How do you know what good eye movements are? Right? It's kind of difficult conceptually. Like how do you know where people should be looking? Right? So for this, what we looked at is what we call typicality. So we have 24 movies. They're all social. We looked at, and we piloted 60, and we chose the 24 where the eye movement patterns, the gaze patterns, were the most consistent across participants. So this is an example of a movie clip. These are very short, like 10 seconds.
How can we even be sure she's going to help us free the childlike empress? I do not trust her. I can tell when someone lies to me. I can tell when someone is up to no good.
Okay, so that's like one example. This is a second example.
You find it odd? Your predicament?
Oh no. Can I leave now?
No, you can't.
Okay, so they see 24 of these, and we do eye tracking while they're watching them. We had 62 typically developing participants and 36 participants with ASD. And this is what the eye movements look like. These are a single subject, right? So each dot here is a subject. One group is the TD group, and one group is the ASD group. They watch them with sound, of course. Okay, so usually here I do like a show of hands who thinks the blue group is the ASD group and who thinks the red group is the ASD group. And usually people answer about 50-50. It's very difficult to tell just from looking at this. It's very difficult to tell which would be the ASD group and the TD group, because on average, like generally they're looking in the approximately the same. Okay. This is another example. Okay, I hope you can see from this that the least typical subject is not inattentive, right? It's not that they're not watching the movie. They're just looking they have different looking patterns than the average, right? So the average tends to be very focused on the center of the face, and people who are less typical tend to have like more saccades, look around a bit more, but it's not that they're not attentive. So this is just showing that like I said, that the averages are very, very similar. And this is true for the TDs and the ASDs. They're very similar to each other. But the other thing that's interesting about this is that typicality is a stable personal trait. So how typical your eye movement patterns are is something that's consistent across different movies. So this is, again, just looking at one particular split. This is average movies, average in the odd movies versus average in the even movies, average of your gaze typicality across all the frames in one half of the movies. And each dot here is a participant. And what you can see is that people who are very typical are always very typical. People who are very atypical are always very atypical. So this is very consistent within participant. It's a stable trait. And the other thing is that the ASD participants on average are much less typical as a group, although some of them are very typical, right? So on average, they're less typical. And also this is what we find in ASD, in all studies of ASD, they're much more variable. Right? So within the group, they're much more variable. They're also more variable within participants. They need more data to be stable. Okay, and this is just showing that this is very significant. Okay, so we have this eye movement typicality measure, which is objective. It has sufficient variance. It's very stable. Maybe it's sensitive to change. We don't know yet. And now we want to find neural correlates. So for this, we put them in the scanner, we put them in the fMRI, and they watch a 9-minute movie, a completely different movie, okay? Because now that I've shown that eye movement patterns are stable, eye movement typicality is stable, then it's stable across all different movies. So now they can watch a completely different movie in the scanner, and we can look at their neural typicality in the scanner. So neural typicality is kind of a modification of intersubject correlations. It's basically for each voxel, for each individual, we can ask how correlated is that voxel to the average activity of everyone else while they're all watching the same movie, right? And these are the kind of maps that we get. They're very similar to ISC maps. And now we can ask, how does the neural typicality relate to the eye movement typicality? And what you see here in blue are all the regions where the eye movement typicality was significantly correlated to the neural typicality, right? So all of STS here, these frontal regions, precuneus, they all show very strong, significant correlations between the neural typicality and the eye movement typicality across participants. Right, so the more typical the neural activity here is, the more typical the eye movement gaze patterns are, right? So what's interesting about this is that these aren't motor control areas, right? This isn't eye movement areas. This is something else. This is something about maybe social orienting or attention, like how they orient attention is more similar. So it means that this is something meaningful that we can look at, right? The red is showing something different. The red is showing group differences in neural typicality between ASD, and nothing to do with eye movements, just group differences in neural typicality between the TD group and the ASD group. And you see that the biggest differences in neural typicality are in the theory of mind areas. So here in TPJ, this is like a classic theory of mind network. Okay. So now we said we want to deconstruct these complex texts, and I haven't shown you anything about that yet. So if we think about the faces again, we see that there are lots of processes involved. And we were lucky to find them when we were correlating resting state with performance on the Cambridge, but really we would like to localize this a lot more. So that was an example from face memory. And now we're moving to social cognition, which is related but different. And here the question is there a social brain, right? Is there something special about social information that's shared across all different modalities and all different tasks? Kind of like the way color is processed in no matter what the task context is or what the, well, it's modality, it doesn't matter what the task context is, you always have the same areas processing color. So is there something like this, like a similar neural mechanism that processes social information across all different domains? And if so, can we isolate the social component of different tasks? And this is work by my other talented grad student, Michal. And the idea is basically this. This is our general framework, right? We have individual differences in behavior, and we try to find the networks, and we try to perturb them. So for finding the first stage of finding these individual differences in behavior, we developed this task battery, which has 4 different social tasks, and each task has a non-social version. And I'll show you the tasks in a second, but the idea is that if there's something special about social information, if there's a shared mechanism that processes social information across all different domains, then these tasks, performance on these tasks across the same people, should be more correlated than across these tasks. Because these tasks are very different from each other. I'll show you them in a second, but they all share this social information. And these are all non-social and they're just different, right? So we have, the first is the movie viewing task. So this is very similar to what we did before, except it's in Hebrew because we're doing it in Israel.
מה תרצי לדעת?
למשל, מה אתה מתכנן לעשות אחרי החתונה?
אני מקווה להיות עוזר בלן שני במקווה של סטנר.
This is Shtisel. You can watch it on Netflix in English as well if you like. We also we show them these social movies. They see 60 of each category. They see 60 of these short social movies. We show them 60 animal movies. Okay, and then they watch another 60 object movies. Okay, and what's shared across all these movies, I hope you noticed, is that it's very clear where you should be looking, right? So we chose movie clips where they're all very directed in a way, right? It's very, and we also piloted this to check the average gaze location in all the movies is also very stable. So on average, people look at the exact same space, and that way we can ask about typicality, because if there's no common average, if the mean gaze location is not stable, then the deviations from the mean are meaningless, right? But if we have a stable mean, then we can start asking about typicality and about deviations from the mean. So we replicate again this correlation between this stability of the typicalities in the social movies. So these are individual participants watching the social movies. This is like now 30 movies and 30 other movies, right? And each dot is a participant. And you can see that it's very, very stable. So people who are typical are always, these are atypical. People who are atypical are always very atypical. People who are typical are always very typical across different sets of movies. But these are all social movies. This is also true for the object movies. It's a stable personal trait for the object movies as well. But if you now compare social versus object movies, then the correlation is much lower, right? And this is quantifying this across all different categories. So within social movies, typicality is very, very stable at the individual level. Within object movies and within animal movies, also very stable at the individual level. But between categories, it's much less stable. So what this means is that basically you can be very typical when watching a social movie and then less typical when watching object movies, or vice versa, right? So your typicality is not the same between the categories. And we see this most strongly when contrasting the social and objects movies, but there's also slight differences when contrasting social animals and animals objects. These also appear to be different, but the most different are social and objects. Okay, that was the first task. The second task is an emotions rating task. We wanted something that's a completely different modality. So it doesn't involve vision at all, but has social and non-social information. So in the social version, they hear these short clips, like about a minute. They have like full context and like a whole story develops. I'll show you just a little bit. This is also in Hebrew, sorry.
לא, א', עכשיו היא קצת אחרי שהיא בקורונה ירדה במשקל. ובעיניי נראית נפלאה עכשיו. יש לה פתאום נתנאים, אני חושב שאף פעם לא היה. אני רואה איך העיניים שלך נהיו פתאום מלאות אור כשאת אומרת. כן, כי קשה לך לראות את הילדה שלך שעומדת על במה עם קבוצה של 20 ומשהו אקדמיות.
Okay, this is a terrible conversation between the mother describing her daughter's weight and how it changed over COVID. Never mind. But I hope you could hear, even if you don't understand Hebrew from the prosody, there's a lot of emotion involved. And then
עכשיו היא קצת אחרי שהיא בקורונה ירדה במשקל. ובעיניי נראית נפלאה עכשיו. יש לה פתאום נתנאים, אני חושב שאף פעם לא היה.
Okay, so then from these from these long clips, we take just one sentence, like a short, like 5-second clip. They hear it again after hearing the full thing, and then we ask them to rate the emotions. Like we said, what did the woman say? How did the woman feel when she said suddenly she has a waist? Right? And then they have to rate all these different emotions from 1 to 7. Okay. And we do something similar for sounds. So this is the non-social version, has no emotions, but it's a complex soundscape. So they hear something like this. I don't know if you can hear it. There's like birds and dogs and chickens and the kids, and they have to rate the intensity of each of these sounds. Okay, and again, they hear a long clip, so it's the same format. They hear a long clip and then they hear a short section from it, and they have to rate the intensity of the sounds. Now, I was very skeptical about this working, that people would, because like I said, we need the average. If we want to look at typicality, if we want to look at distance from the average, then the average has to be stable. And I thought people would never agree either on the intensity of the sounds or on the intensity of the emotions, because these are like very complex. But actually, we found like astounding agreement between people. So this is across, I think, like maybe 40-something people. So it's in each group, it's about 20-something. So averaging across 20-something participants was already enough to get these like amazing correlations in the ratings. All right, this is for the emotions. So each dot here is like one particular question, like what was the emotion of what was the rating of the embarrassment in that particular clip? And this is for the sounds. So we get like really incredible correlations. People really agree on what the intensity was, both of the emotions and of the sounds. So now we can ask about typicality and ask, okay, so if people generally agree what the rating was, and how typical is your rating? Like, are you below, are you above? How are you in relationship to that to the average rating? And what we find is that, again, we get this very broad range of typicalities. Like some people are always underestimate, some people always overestimate, but it's very, very stable within categories. This is for the emotions, this is for the sounds. And so you might say, well, maybe this is just rating strategies, right? Like maybe some people always say 1, 2, 3, and some people like use the full spectrum, and maybe some people always rate highly and some people always rate lowly, and this explains all this variance. But if that were the case, then you would also expect, if you compare emotion ratings and sound ratings, that they would also be very stable, right? If you always rate 1 to 3, then you always rate 1 to 3, right? And then there should be no difference in the stability of the typicality for emotions versus sounds. And that's not what we find. We find much lower correlation when we contrast emotions to sounds. So again, this means you can be very typical on the emotions and atypical on the sounds, or vice versa, right? So this summarizes this. And the third task is a motion inference task. So this is actually adapted from work by Mariam Vaziri Pasham, who did this in Harvard a while ago. She did something very similar. So this is based off her work. And what we do is these are students of mine that we filmed, and they were told to either go left or go right. And we show only the first frame of motion, basically, or the first few frames of motion. And people have to say, after this, they see a blank screen, and people have to say, is he going to go left or is he going to go right? Okay. And we have a few different demonstrators, and they see, I think, about 100 trials of these. We also have people jumping. So they're going to jump either left or right, and you only see the beginning of the motion. And for the non-social, we have this kind of physical motion. So we have like a mobile, and you have to say, is the wind coming from, is the wind blowing left or is it blowing right? We have the mobile, and we also have leaves on water, right? And there again, like in the previous tasks, we find that there's a lot of variance. So some people are a chance, some people are perfect. We get like pretty much the full spectrum in between. So there's a lot of variance in how well people are on these tasks, how well people do on these tasks. And it's very stable. So if you are good, you're always good. If you are bad, you're always bad. And the social and the non-social ones are different. So you can be here, like this person is super accurate on the social and was like completely wrong on the non-social here as well, right? Like all these people they do very differently on the social versus non-social. So there's a very low correlation between them. So basically what this means, all of these differences means that we're able to behaviorally separate these different processes, which indicates that there must be a different neural mechanism, right, guiding them. So for the movie watching, we know this is true because we know that social cues are different from saliency cues, for instance, and that social cues are used in social situations, like when you're watching a movie, on top of saliency cues. So we know there are different attentional mechanisms. For the motion prediction as well, biological motion is known to be different from non-biological motion. So it's maybe not surprising that these are different, but this is a really good behavioral measure for separating them. And now once we have this, we're able to put them in the scanner and look for the neural correlates. So this is basically where we're at. We see this convergence of each of our tasks. So there's a stable mean to each of the tasks. We see variance between participants, but stability within participant on each of the tasks. And we also see differences across categories, meaning that these tasks are different, like they have different mechanisms. So now the big question is what happens across tasks, right? Because we started with if there is something shared if social information is special in some way, and then we expect that performance on the social versions of these tasks will be more similar. So we don't actually find that. We don't find something that's similar across all the different social tasks. What we do find is that the empathy task or the emotion rating task is very well correlated to the social motion, and also slightly less so, but still correlated to the non-social motion. So it means that the more typical you are on the emotion ratings, which we assume, this is an assumption, but I think it's true definitely for the emotions, that in a way, being more typical is like being more optimal. Right? It's like if you are, if you think about, say, people driving along a racetrack, so they will take different trajectories along the track, but the average trajectory will be optimal. And the most typical person might be like a Formula One driver, would be very close to this optimal trajectory, and it would be very typical. So I think this is true, especially for the emotions and maybe also for the eye tracking. And so what we see is people who are very typical on the emotions, or at rating the emotions, are also very good at predicting the biological motion and also at predicting the non-biological motion, but even more so on predicting the biological motion, which is nice. Okay, so I said what we do is we characterize behavior and individual differences. So we're pretty much done with that stage now for this project. Now the goal is to find the underlying neural correlates. This is where we're at right now. So we're looking for the brain regions whose neural variance predicts the behavioral variance that we see across participants. This will allow us to form an hypothesis about which networks are involved in which behaviors of which different tasks or processes. Then we can directly perturb these networks with covert neurofeedback and then assess the change in behavior. Okay, so I'd like to leave you with this take-home message from Life of Brian. You are all individuals, right? And we see, this is, I think, one of the main, truly the main takeaways from this work is we see so much variance across individuals in everything that we measure. People are really very different from each other, but consistent within themselves. And to see this consistency, though, you need to have enough trials. Like, you need to measure enough. But if you do, then people really are very consistent. And thank you. This is thank you to my group and to especially to Michal Zamberg and to Jan Kadris, whose work I presented here, and to my funding sources and to our collaborators. And thank you all for listening.
Michal, thank you so much for a really exciting talk. I want to invite everybody here to unmute yourself and let's give Michal a big applaud. Really exciting work. And I'm opening the floor for questions.
Should I stop sharing so we can talk maybe?
I see that whatever you like.
Okay.
Well, I actually have a question, one of many. You said that, for example, the average would be, you think that, you know, the like Formula One driver would be, you know, near the average, but then we are sometimes, you know, but then the average that you're describing is not being mediocre, or so it's,
yeah,
I
it's,
mean,
it's not average.
so,
like average is maybe misleading in that sense, like the term.
Because you think all these, let's say, individual variances from the mean are actually a kind of like some interference of leading them to perform perfectly, I would say.
Yeah, I think it's just a different construct maybe, because when we think of average, like average on a test, like how if you average how well everyone did on the test, and that would be average. And if you're closest to that average, you are an average person, right? You're not like the best, you're average. But this isn't like scores. We're not averaging scores on a test. We're averaging, so it depends what you're looking at when you look at typicality. But if you're for something like this where it's you know, either the ratings on the emotions and the sounds or the eye tracking, like the average of where people look is averaging across all the noise in a way. So you're left just with the signal. And that signal is like the optimal, maybe.
Yeah, this is fascinating. Really, it's another level of thinking about things. I want to ask if you, I mean, there are things, for example, there are paradigms that you can run where you can use the same physical information but manipulate the task. I mean, you probably, you know, the Yarbus, where he showed those same pictures, but he gave different tasks, you know, what is the social status, what is the connection between the people. And this really changed the way people looked at things, or let's say we know how task influences everything. So do you think this, even if you leave the physical stimuli the same, but you modify the task, you would then examine within individuals differences in their behavior, but consistency across themselves for a specific task, which would probably tap into different processes, even given the same physical, do you understand what I mean? While keeping, so I'm just wondering if you, because it's probably not the physical aspects of the task that drive these individually consistent behaviors.
Yes, that would be my guess, that if you give it, so for instance, for the movie viewing, if you gave a task that had them scanning differently, then I think what would happen is you would see different averages, like the mean would shift. And then your typicality would also shift in the sense that now you're measuring something different. So it's not how, if you're not giving any instructions, then you're basically looking at basic attentional mechanisms driven by the cues in the movies, right? The saliency and social cues and whatever in the movie. If you give them a different task, then now you're looking at different mechanisms. So your typicality won't necessarily be the same, but I think it will be consistent.
Interesting. And I see still no hands raised, so I've got another question. You said you saw in the Cambridge face memory test that you know, people or improved, and this was so what it was, did you, or you know, the fact that you provided some neurofeedback that they did not have any idea what came from, but did you interview them, or do you think they may be aware of the training or the changes, or were they?
We interviewed them. They had no idea what the training was about. They didn't know they were being trained also. But they also didn't feel like they had improved in the face memory, which is also interesting. So they had no, they didn't say, oh, but suddenly I remember faces so much better. I don't know even if that's something you would necessarily notice about yourself over a week or even like a few months. Maybe I think I think it's very difficult to change our perceptions of ourselves. So if you, especially if you have no conscious reason to change them, like it would be interesting if we told them we were training them, would they then think that they got better? But they didn't think that they got better, but they had no idea what the training was.
I see, interesting.
Also, it was
I mean,
different.
maybe also, yeah, sorry.
It was different forms of the Cambridge, so it's not the same faces.
Oh, okay, brilliant. That's really great. So it's even more convincing. I'm just wondering, maybe it's also because the change was gradual. Maybe that's also why they haven't noticed that that it changed, because it was some, you know, I mean, slowly. It's not that it was like one big jump in their performance in like a day. Because you showed that it was, you know, building up over 4 days. So maybe then it's
Yeah,
really
maybe.
interesting.
Maybe. I think it's more that it's very difficult to change perceptions of yourself, but it would be interesting to see if there was like a large shift, if they would notice it, like a sudden shift.
Yeah. Did you try to do it for prosopagnostics or people who have face perception difficulties to see if they improve, and if they do improve, does that in a way reach their awareness?
They're kind of like borderline prosopagnostics in a way. They're not, they're very bad at faces, but they're not classified as prosopagnostics. But maybe Galia and I will look at this in the future.
Oh, lovely. I see Galia is here.
Yeah.
Great. Yes. I see Hagit and Hamutal have questions. I don't know who raised their hand first, so just unmute yourself and go.
I can go first. Okay.
Go ahead.
So Michal, thanks a lot. Really a great talk, very interesting. I think I have 2 questions. There was I was wondering about this. So we are all, as you said, that the sort of like the take-home message is that we are all, or you're all, individuals, right? But within an individual, if I got it right, you have constant behavior, more or less. And so I was wondering, there was a recent paper by Elisha Merriam and Zvi Roth about representation in human primary visual cortex, the drift over time. So what they were suggesting is like that the brain representation of some, you know, stimuli in that case are actually changing over time. And so I sort of like, I was wondering whether it's possible, or do you think that maybe individuals also show within themselves drift across time? We are working with monkeys, and what I see, at least in monkeys, which are not humans, I know, so maybe they are much less consistent, who knows? But what I see in monkeys is that we measure for monkeys for you know, several months and sometimes for several years. And what we see is that they can change their behavioral sort of like attitude and strategy for the same performance over time for some unknown reason. But maybe this is unique for monkeys, also in terms of the eye movement. So this is my first question.
Okay, so wait, so let me answer this question because otherwise I'll forget.
Okay.
So my guess actually would be that monkeys are equally consistent. And to qualify this, of course, I'm not saying that people can't change it, that people don't change it, that they never change. Like people learn and they learn new things, they can do new things that they couldn't do before, and and they get better, they get worse. People change over time. This was more within a given time point, more or less. Actually, you know what? Let me share again, because I have this slide that I wasn't going to talk about. So this is part of the reliability project that we did. And what I showed you before was the split halves reliability. So we had people they came for like 4 different testing days, and sometimes we had testing days that were further apart as well. And in what I showed you before, we treated all the data as coming from the same time point. So like all the data was the same. We randomly split it into 2 halves. Here we looked at time. So this is like more classic test retest. So we have test in one time point and retest in another time point. And before I told you that everything converges to 1. Like in the end, if you have enough trials, the reliability converges to 1. But that's if you look at everything as if you mix everything as if it's coming from the same time point. If you now separate these into different time points, then this is what it converges to. This isn't the reliability. This is the reliability it converges to. And what you can see is that for most tasks, it doesn't converge to 1. So across different time points, people are different, right? So, and it's different across different tasks. So some tasks, we hardly see this. These are the more, like the simpler and more perceptual tasks, where the tasks that have more working memory or more attention, we see greater different, more like memory, we see more differences across time. But if you average, I don't have this for all the tasks, but for one task that we looked at, we had like 6 different forms, and they came in over a month. So you had like 2, and then a week later they had another 2, and then a month later they had another 2. And if we average, so this is what happens, this is how it converges if you look at just one day versus like one single day versus another single day. And you can see this isn't going to converge to 1. But if you average 2 or 3 days, across 2 or 3 days, then they do start converging. Right? So you are different. You are not the same person between different time points, but you're kind of fluctuating still around the same mean. So if I now take two days and 2 other days, then those are already quite similar to each other.
So I guess that would also affect the analysis you're doing in terms of correlating it with brain activity, right? Because if an individual actually varies a little bit in its behavior, then maybe you expect also the brain activity changes a little bit. And if you're looking to the resting state mode or things like that, maybe that also varies with that within this individual. So
yeah, so what we're trying to do now is to really test across 2 or 3 days and then average. So then you get this more stable pattern that's even stable across days. So that's and also, but just what you said about the representational drift, there's Yaniv Ziv also has work on this. So I think this is definitely a real thing, but the representational drift doesn't mean behavioral drift. I think these are 2 separate things. So you could have representational drift, maybe because what you're calling representation isn't really representation. Maybe that's not the true representation, right? There's like the whole debate about this, but it doesn't mean that the behavior is changing necessarily. So that's like a side note.
I think I'll let Hagit ask a question if we have time, or later on I'll ask again. Thanks.
Thanks.
No, my question is very kind of basic. But first of all, thank you, Michal. It was an amazing talk. I really, really enjoyed it. Thank you. I wanted to ask about the movies. At least the ones that you showed, they sort of naturally have one central focus area. And in many of the social interaction clips that are used, especially in the ASD, there's more action going on, or more, let's say, more centers of attention or possible attentions. And I was wondering if you looked at these kind of clips, and then, of course, the actual position of the eyes, averaging over them, will have to be sort of clustered, and maybe correlate the number or the positions of the clusters rather the actual eye positions. And that goes together with the study about how I actually think it's neurofeedback as well that trains ASD to focus their view location, their eye tracking, to more social oriented locations. And it has shown that they are able to learn and train. And then I'm thinking it could be really interesting to see how that works in the fMRI.
Yeah, so for the multiple gaze, the examples I showed really have one, actually one had two people, but the examples had mostly one person, but we show 60 clips, so about half of them have more than one person. And some of them have like some of them are like more party scenes and there's a lot going on. Some of them are like one particular person, but we have a full range. We also looked at, does it matter whether, so what happens there when you're at, like what you said about the averaging is you'll have some people look at one person, some people look at the other person. Sometimes it's very clear. Sometimes like one person is talking and you look at them. And sometimes you look at like half and half and it doesn't have to be consistent. But on average, it's still consistent. Like on average, there's a bias towards one or the other. So we looked at, in terms of the analysis, whether it makes a difference if you compare it to the average or you compare it to the like a hotspot, like a local hotspot, and it's the same, it doesn't matter. Like it makes no difference. And about the training, what's actually really interesting is in the study I showed you with the social eye movements that we published with the neural typicality, we also have another paper where we looked at, for the ASDs, especially like where they were looking at. So we had labels on each pixel, like what it was, like eyes, nose, face, whatever. And we found that, contrary to popular belief, so the ASDs definitely look at background more. They look less at faces in general. But when within the face, they look slightly more, like not significant, but close to significant, more at the eyes than the TVs. And I think this might be because they're trained to do it, because a lot of them are like taught to look at the eyes. So yeah, so I think definitely you can teach them to look in particular locations because it seems to have worked for these.
Right. But then it would be interesting to see where in the brain this training, where it's affecting, right?
Yeah. So what we found for the eye movement typicality was that we got the exact same regions for the TDs and the ASDs that were correlated with the eye movement typicality. So it's these regions of like STS. So what we're looking at now that we have the social versus non-social, we'll be able to see which regions are specific for social cues versus like more salience cues or whatever. So then, and maybe those will be particularly important in ASD or.
Cool. Well, I hope you come back and tell us the results.
Yeah. So
thank you.
Thanks.
Definitely. So Michal, I want to thank you a lot for joining us and for a really great and insightful talk. So thanks again. Next week, we will not be here. There is there's
Memorial Day. Memorial Independence Day.
But we will post our next seminars. I'll send an email about them. So thank you, Michal.
Thank you. Thank you for having me.
Thank you. It was a great pleasure. Michal, I want to ask you, my internet connection was it had issues. So if you are happy to, wait, I thought I stopped recording. Did I not? Yoram is recording. Okay, Yoram. No, that's good.